In the first phase of the cell cycle, interphase, a nuclear envelope encloses the nucleus. Two centrosomes have formed by a duplication of a single centrosome. The chromosomes, which were duplicated during the S phase, are grouped together and uncondensed. In the second phase of the cell cycle, prophase, chromatin fibers become more tightly coiled. Each duplicated chromosome appears as two identical sister chromatids joined at their centromeres. The mitotic spindle begins to form, composed of centromeres and microtubules. These extend from the radial arrays of shorter microtubules. The centrosomes also begin to move away from each other. In the third phase of the cell cycle, metaphase, the nuclear envelope dissolves into tiny fragments. The chromosomes become even more condensed. Each chromatid now has a kinetochore. Also, some microtubules attach to the kinetochores, causing the chromosomes to jerk back and forth. In metaphase, centrosomes are now at opposite ends of the cell. The chromosomes line up at the middle of the cell, forming a metaphase plate. This plate is equidistant between the spindle's two poles. Also, the chromatid's kinetochores are attached to the kinetochore tubules coming from opposite ends. During anaphase, the two parts of the sister chromatids separate from each other, and each chromatid now becomes a chromosome. These chromosomes then begin moving toward the opposite poles of the cell. While this happens, the cell elongates. Finally, two ends of the cell have the same number of chromosomes. In the last phase of the cell cycle, telophase and cytokinesis work hand in hand. The cell forms a cleavage, which means that the cytoplasm pinches in two. Also, two daughter nuclei form. The chromosomes in the nucleus once again become less condensed and look almost like noodles. The yellow spindle microtubules also disappear. And there you go. Mitosis is now complete. Two genetically identical daughter cells are formed.
Interphase 1. The cell prepares for cell division by replicating its chromosomes. Each chromosome has two sister chromatids held together by a centromere. In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapse and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach only to one side of the centromere and the two homologous chromosomes attach to microtubules orienting from opposite poles. In anaphase 1, the microtubules on the spindle fibers shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. Telophase 1. The nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains sister chromatids for each chromosome, attached by a common centromere. Because of crossing over, the two sister chromatids are not identical. By cytokinesis, the cells cleave apart, creating two daughter cells. In prophase 2, the nuclear envelope breaks down and new spindle forms. In metaphase 2, the chromosomes line in the center and the spindle fibers bind to both sides of each centromere. During anaphase 2, the spindle fibers contract and the sister chromatids move towards opposite poles. In telophase 2, the nuclear envelopes reform around the sets of daughter chromosomes. By process of cytokinesis, the cells cleave apart, creating 4 cells.